Hello, everyone, and welcome to today's webinar. My name is Catherine Heldy, and I'm the Chief Scientific Officer here at Cure SYNGAP1, formerly known as Syngap Research Fund. I have the pleasure to introduce today's speakers, Dr. Megan Quinlan, PhD, and Dr. Rong Guo, PhD. Megan Quinlan joined the Allen Institute as a therapeutic lead on the AAV gene therapy team in the human cell group in 2022. As part of the gene therapy program, Megan designs and tests new therapeutic AAVs to treat brain diseases such as epilepsy. Prior to joining the Allen Institute, Megan researched the impact of disease-associated gene variants at the molecular, neural circuit, and behavioral levels. As a postdoctoral fellow at the University of Washington, she studied potassium channel variants identified in neurodevelopmental disorders and worked on developing a novel cell type specific CRISPR-Cas9 gene editing technology. She got her PhD in pharmacology at Vanderbilt University, studying the structural and functional dynamics of serotonin transporter gene variants identified in autism spectrum disorders. Megan received her Bachelor of Science degree in psychology with a minor in pharmacology and mathematics from the University of Vermont. Currently, Megan is a project lead in the development of AAV-based gene therapies for neurological disorders at the Allen Institute. Her background is rooted in molecular biology with a strong focus on neuroscience and pharmacology. I have had the pleasure of meeting Megan in person a couple of times. Most recently was after I had seen her and met Rong for the first time at the ASGCT, so the American Society of Gene and Cell Therapy meeting that was in New Orleans this past year in 2025, where I heard her talk and it was amazing. And so that's what got us talking. And I live in Seattle, and these amazing scientists work in Seattle. And so I got to go down and see the Allen Institute for Brain Science, which is right near where I live. So that was great. So now I'll tell you about Rong. Rong Guo, PhD, is a scientist 3 at the Allen Institute for Brain Science, also in Seattle, Washington. Her research focuses on developing molecular, genetic, and circuit level therapies for neurodevelopmental, neurodegenerative, and psychiatric disorders. She leads a behavioral and in vivo electrophysiology pipeline to evaluate novel adeno-associated viral, which is AAV, therapeutics for the treatment of neurodevelopmental epilepsy, which SYNGAP1-Related Disorders is our top neurodevelopmental epilepsy for this crowd. So before joining the Allen Institute, in 2022, before that, Dr. Guo was a postdoc associate at the University of Pittsburgh, where her work used multidisciplinary approaches to understand how sleep regulates reward seeking and relapse. She obtained her PhD in neuroscience from Washington State University, go Cougs, where she studied sleep and circadian disruptions in rodent models of alcohol use disorders. She received her BS in biological science from Shandong University, China. So she has a lot of current work, including things with Dravet syndrome, SYNGAP1, and SLC6A1 related disorders. And my most recent conversations with Rong had to do with EEGs. She's has incredible expertise in mouse EEGs. And we're really lucky to have her working on this, and her goal is to bridge basic neuroscience discovery with translational gene therapy approaches that can bring meaningful treatments to patients with genetic epilepsies. So just a little bit of housekeeping before I turn it over. A recorded version of this webinar will be available on our website at curesyngap1.org. You can find the webinars tab under the news and events menu. And at the end of the presentations, we will take questions from the audience. We'd love to hear from you. Please write your questions in the chat or the Q&A. And for those of you just joining us, welcome. And again, our speakers today are Dr. Megan Quinlan and Dr. Rong Guo. of the Allen Institute, and their presentation is titled AAV Gene Therapy for SYNGAP1-Related Disorders. And with that, I will turn it over to you guys. Thank you.
Oh, well, thank you so much for that really kind introduction. And I want to give a special thank you to both Mike and Catherine for inviting Rong and myself to give the webinar today. As she mentioned, we're both scientists at the Allen Institute here in Seattle, and together we co-led this effort on generating an AAV gene therapy for SYNGAP1-related disorders. And our goal today is to really walk you through what that actually means, right? What is an AAV? How does gene therapy actually work, and how did we apply that specifically to SYNGAP1-related disorders, or SRD? And so to start, I like to start with this cartoon that one of our colleagues actually drew, M. Luber, who is also on our team. It's just a DNA and a therapy session, which I think is kind of cute. Because I know gene therapy can kind of sound complex and intimidating, but at its core, it really is a really simple idea. We're trying to give a healthy gene to fix a disease caused by a loss of that gene, right? So today I'll show you how we used AAV, which is just a type of delivery vehicle, to give the SYNGAP1 gene back to try to restore SYNGAP function in the brain, and then ultimately, hopefully, cure SYNGAP1. So our goal isn't just to show the data, but to make sure everyone leaves with a clear understanding of what we mean by AAV gene therapy, and how we used it to test potential treatments for SYNGAP1-related disorder. But of course, if for anyone who wants to dive a little deeper into the science itself, our full paper is published open access online in Molecular Therapy. I provided a QR code, or if you just search online AAV gene therapy for SYNGAP1, this should pop up for you. But first, we also have to share disclosures, because it's important to be transparent, especially when discussing potential therapies. So this project was funded in part by BioMarin Pharmaceuticals, but every experiment and all the materials were generated and performed at the Allen Institute, where our team led the research. So, and we are also listed as inventors on a patent derived from this work. So to start with today's talk, I want to actually give a quick funny story about how I first connected with the SYNGAP1 community, because I think it really shaped how we think about our work towards a possible therapy. It was actually on a flight that I happened to be sitting next to Catherine Helde, the now CSO of Cure SYNGAP1. We are both from Seattle, as she mentioned, and I was heading to my first conference for the SYNGAP1 Research Fund at the time. And I had just joined the Allen Institute and started working on this project, but we were still under a non-disclosure agreement, so I really couldn't share anything yet of what I was working on. But I remember Catherine finding out I was a scientist, and she was so excited and was trying to convince me to work on SYNGAP1. She shared stories of her son and their daily challenges, and she really emphasized the real urgency that families were facing, right? And I got to go to the SRF conference and meet even more families there. And hearing the lived experiences really brought the science to life in a way I hadn't experienced before. And I quickly realized is that the community does more than just raise awareness. It actually drives science forward, right? I wasn't originally working on SYNGAP1 before I came to the Allen Institute, but because of Cure SYNGAP1, right, the webinars, Mike's podcast, and even Catherine's book, which I read, we were all able to get up to speed really quickly in the SYNGAP1 space and begin making meaningful progress. And that's rare in science. So in many ways, the reason we're able to talk about a gene therapy today is because of the community that makes it possible. And that brings us to our starting point today, right? What is SYNGAP1? I know everyone here knows what the disorder looks like, right? You know, it's epilepsy, intellectual disability, sleep disturbances, risk behavior. I don't need to explain the symptoms to this group because I know you experience them every day. So instead, I want to focus on not how SYNGAP looks, but actually focus on what causes SYNGAP1-related disorders and why those symptoms happen. And that's because of pathogenic variants in the SYNGAP1 gene. And understanding that really leads us to gene therapy itself. So pathogenic variants are simply just harmful changes in the DNA sequence. They disrupt the SYNGAP gene in a way that prevents the brain from making a full or functional SYNGAP protein. So every gene has two copies, one from mom and one from dad. And here on the left, you can see I have a healthy copy of the SYNGAP1 gene, or the DNA sequence made of like A, G, C's, and T's. And that gene makes a healthy SYNGAP protein. But on the right, we have a harmful variation, or sometimes what we call a mutation, that can occur either spontaneously or be inherited. And this variation will produce either an abnormal SYNGAP protein, or we'll get a complete loss of the SYNGAP protein. And a healthy brain needs both copies of the SYNGAP1 gene to make enough SYNGAP protein, because one simple copy is not enough. So in this case, the brain ends up just with not enough SYNGAP protein. And this reduction drives many of the symptoms that patients experience. So from a biology standpoint, if the problem is not enough SYNGAP protein, can we give a patient a healthy copy of the SYNGAP1 gene so that it can produce more SYNGAP protein? But of course, I can't just hand you the SYNGAP1 gene. So how do we actually deliver a healthy copy of the SYNGAP1 gene? So in order to do that, to get the healthy copy into the brain, we as scientists use something called AAVs, which stands for adeno-associated virus. And I know now when most people hear the word virus, they naturally think of something that makes you sick, like the common cold. That's completely understandable. But AAVs that we use for gene therapy are actually very different. In fact, as a human population, most of us have already been exposed to AAVs, and they rarely make us sick. And to make them even safer for therapy, scientists have actually removed all the viral genes that could make you sick or allow the virus to replicate and spread. So what is left essentially is just a hollow virus shell that can act like a tiny little delivery vehicle for DNA. And viruses are extremely good at delivering DNA specifically to cells, because that's what they do. So just like a little car, the AAV's only job is to carry a piece of DNA. In our case, that's a healthy copy of the SYNGAP1 gene into cells, and ultimately into the nucleus, right? Where the cell can actually be processed and produce more SYNGAP1 protein, which is the most important part, right? And then for the SYNGAP1 gene therapy, the cell we actually want to reach are the neurons. Those are specific cells that are located within the brain. And once the AAV gene is delivered into the neurons, it stays there, and it helps make more SYNGAP protein over time. So the AAV doesn't multiply, it doesn't spread, doesn't do anything beyond delivering the cargo, and so that the DNA can continually make more SYNGAP protein. And that's why we consider AAV gene therapy is designed to be a durable, long-term treatment lasting years to decades. And now that we've talked about how AAVs are delivered, let's look at like what did we actually put inside the AAV itself. So here I'm depicting our actual gene therapy cargo. That's what's inside the AAV shell or capsid. And it has several key components that I'll walk you through. First, the most important part of what we're delivering is the SYNGAP1 gene itself. You might remember that the SYNGAP1 can produce several different versions of the SYNGAP protein, and we call those isoforms. And these isoforms can differ depending on where the gene starts, or also at the end where it gets spliced out differently. So the big question is, which isoform do we actually deliver? Because pathogenic variants can occur anywhere along the gene, we decided to choose the longest N-terminal isoform, which is A. This ensures that our therapeutic construct also included all the gene's potential functional domains as well. And for the C-terminal end, so that's at the end here, we selected the alpha-1 isoform. A lot of research has already been done in this space looking at alpha-1. And it's been linked previously to reducing seizures and related neurological phenotypes. So that made it particularly relevant for our gene therapy specifically. And then just before the SYNGAP1 gene, we actually also added a critical component called the human synapsin 1 promoter, or HSYN1. You can think of a promoter just kind of as the gene's on switch. So it makes sure that the SYNGAP gene can only make SYNGAP protein, primarily in neurons and not in other cells where SYNGAP is not needed. So the promoter is really important to make sure the gene gets made into protein. And then just at the other end of the SYNGAP1 gene, we also added a 3X flag tag. You can think of this just kind of as like a small label that we attach so we can visualize and measure the SYNGAP protein of our gene from our gene therapy and experiments. So it just really helps us track what we're producing in the lab itself. But it's important to know that for any gene therapy construct that could potentially be in humans, this would be removed. It's only used in preclinical studies to track it. And last, we also just add these smaller supporting elements called a WPRE3 and a short poly A sequence, and they help the gene message be processed and properly expressed efficiently within the cell. So they are important, but I won't spend too much time on them. But altogether, these pieces ensure that when the AAV reaches the neuron, that healthy SYNGAP1 gene is turned on and produces the SYNGAP1 protein or the SYNGAP protein in the correct place. But now the question really becomes, how does our AAV gene therapy does it actually work, right? That's the main questions. So to test our gene therapy, we actually evaluated this on multiple levels. We'll start with an in vitro functional assay. These are just experiments performed in cells in a dish, so it's much easier. But then we'll move to in vivo delivery, which is putting the AAV SYNGAP1 gene therapy directly into mice so we can see, does our construct actually produce the SYNGAP protein in a living animal? Finally, and probably most importantly, we'll test to see if our gene therapy can actually rescue disease-relevant phenotypes in the mice as well. So let's start with the in vitro data. So here we're essentially asking, right, does the SYNGAP1 gene that we've made work as we expected in cells? And these experiments are faster, they're easier than testing in mice or animals, and they help us identify whether the construct behaves properly before we're moving into a more complex living animal system. So we first wanted to test the enzymatic function of our SYNGAP, by which it's named for. So as you all know, SYNGAP stands for a synaptic GTPase activating protein. The name reflects two key points, right? One, that it's located at the postsynaptic neuron, and two, it contains a gap domain, meaning its main enzymatic function is to inactivate small GTPases like RAS. So in simple terms, SYNGAP acts to decrease active RAS levels, and that's RAS bound to GTP. So to measure if our SYNGAP construct can decrease active RAS levels, we used a specific ELISA-based assay where we can actually measure the amount of active RAS in cells. All right, and so to do this in cells, and we specifically use these HEC293 cells, which are a type of cell line commonly used in labs. We added RAS, and then we can measure the amount of active RAS in those cells, and that's shown here in the white bar. And then in other cells, we added both RAS and our SYNGAP1 construct. And when we measured active RAS levels here, you can see by the blue bar, we see a clear decrease in the amount of active RAS levels. So this shows that our SYNGAP1 protein produced from our construct retains the gap activity, meaning it can still inactivate RAS as we expected. But for SYNGAP to be effective, it also needs to do its second job, and that's localize correctly within neurons. So to see if the protein reaches the right place in neurons, we actually use rat neurons in a dish rather than in a live animal. So all we had to do here is we can actually add our SYNGAP1 construct directly onto these neurons in a dish. And then we can stain for SYNGAP1 protein. And so on the left here, you can see the green labeling along a neuronal projection of a rat neuron, showing where the SYNGAP protein is actually located on the axon. We also co-stained with PSD95, which is a well-known marker for the postsynaptic density. I know many of you probably have heard about PSD95 because it interacts with SYNGAP. and more specifically, it interacts with the alpha-1 isoform, which is the isoform we have. And as indicated by the white arrows here, you can see that we see some co-localization of PSD95 in red and Syngap in green along the neuron. And so while this isn't a direct measure of interaction, it does suggest, though, that our Syngap is reaching the correct subcellular compartments. And then again, if you remember, we included that flag tag, right, to track the protein. That's depicted here in purple. You can see the flag signal overlaps with our Syngap signal, again, confirming that the way we're tracking this is we're able to detect both the protein, it's the Syngap protein, and the tag that we have. So together, this data shows that our AAV SYNGAP1 construct produces protein, right? And that it reaches the right localization within neurons. And so that's was really critically important. So now that we've seen our SYNGAP1 construct can work in cells in a dish, the next question is, does it work in a living animal model? So to answer this, we move to what's called in vivo studies. So essentially, we're asking, if we delivered the AAV SYNGAP1 to a living animal, can it produce Syngap protein in neurons as we expected? This is a critical step, right? Because the therapy that works in cells in a dish may behave differently in a complex living environment. So, but before we do this, we need actually an animal model of SYNGAP1-related disorder that mimics what happens in mice. So just like humans, mice also have 2 copies of the SYNGAP1 gene. So in normal mice, which I have here on the left, we often call these wild type mice, where we have both copies are healthy and producing a full amount of the Syngap protein. I know this is what Mike often calls them too. You can think of a wild type mouse as an unaffected sibling. And then to model SYNGAP1 related disorders, we reduce Syngap to just 1 working copy, and we do this using molecular scissors called Cre recombinase, where we can actually just cut the gene out at specific DNA sites. So more specifically, at a scientific level, we did this by crossing a CMV Cre mouse line with a flocked Syngap line, and that generated our SYNGAP1 heterozygous mouse, just meaning, though, at the end of the day, this would lead to about half as much Syngap protein. And that mirrors what we see happen in humans with SRD. So for simplicity, though, for throughout the talk, I'll just refer to these mice as an SRD mouse model. So now that we have that, we now need to dose our animal, but we first needed to determine when to deliver it, right? As the timing depends heavily on developmental stage. So here I'm showing a developmental timeline where on the bottom you can see the mouse development, and on the top is the rough equivalent developmental stage that we see in humans. And since mice develop much faster and they don't live as long, instead of tracking their age in years, we track their age as a postnatal day, so that's P. So it's essentially just how many days the mouse has been alive after birth. So it's just the number of days old they are. So for our gene therapy, we decided to focus on 2 key delivery windows. First is actually postnatal day 2, P2, which is comparable to a human neonatal stage. So at this age, we can deliver the AAV directly into the cerebral fluid in the brain's ventricles. This method is called an intracerebral ventricular, which is a long word, so I'll just call it an ICV injection. This is pretty quick and very feasible in very young mice. And importantly, it enables us to get the gene directly delivered into the brain. The next stage we decided to also test was postnatal day P21. This stage aligns more closely with the juvenile phase in humans, which is often when patients begin to receive a diagnosis for SRD. And here, instead of the ICV route of administration, we decided to do an intravenous route, or IV, which in mice we can actually easily do by just injecting just below their eye. It seems like it would be hard, but it's actually pretty easy. We call this a retroorbital injection. And this allows the AAV to circle into the bloodstream so it can actually reach all areas of the brain. But to do this successfully, we actually need one more special component, and that's actually a specifically engineered AAV capsid that can cross the blood-brain barrier so it can get into the brain. In our case, we're using a capsid called PHP.EB, which is just optimized for that purpose in mice. We also tested our vector at 3 different doses that we'll call in our talk low, mid, and high. But just to give you a sense of the scale of this, our low dose is at what we call 1E11 viral genomes, which actually is about 100 billion viral particles. And then our high dose goes all the way up to what we'll call 1E12, but that's roughly equivalent to 1 trillion viral genomes. Of note here, though, I do want to mention, though, for our P2 ICV injections, we did only use the low dose, and that's because at this age, the mice are very small, and once you normalize to body weight, the dose effectively corresponds to what we would see for the mid and high dose that we gave in the P21 animals. So with those parameters, we have now timing, when are we giving it, the route, so how are we going to give it, how are we going to inject it, and the dose that we're going to give. The next question becomes, how well does our AAV SYNGAP1 gene produce SYNGAP protein in the mouse brain, right? So to test this, we look directly in mouse brains itself. And so here you can see 2 examples from each of our injection methods, the P2 ICV and the juvenile IV route of administration. And just to orient you, what you're looking at here is just a slice of a brain. Here would be like the nose of a brain, and here would be like the neck and the back of its head. So that's just it's called a sagittal brain section. And to detect our SYNGAP protein, we're actually going to stain for the flag tag that we added, just so you can, and that's what you see here in black on these 2 images. And on the left, right, we have the P2 using the ICV method. And here we see expression in several key areas, notably the cortex, which is up here, the hippocampus, and the striatum. But I think you can appreciate that the expression is a little bit more focal. It's more concentrated in the cortex rather than across the whole brain that we see. And then in contrast on the right, when we're looking at that really high dose that we gave of the IV and older animals at P21, here we start to see a much broader brain-wide expression of our SYNGAP flag protein. And this is consistent with that virus being able to cross the blood-brain barrier, as I mentioned earlier, and reach multiple brain regions at one time. But now if we actually look just a little bit closer here at the cortex and we get a zoomed in view of what we're actually looking at, we do see we get strong labeling in the cell bodies. It's important to note because if you just stain for what SYNGAP looks normally without a virus there, we typically don't see this kind of cell body pattern of SYNGAP expression. However, when we do zoom in even further, I want to point out we can see some axonal labeling as well. So that's where we want to see SYNGAP be actually expressed. So here I'm staining for both flag, and now it's in purple because you just make it any color you want. And SYNGAP, the protein here, is in green. As you can see, we're getting some really nice overlap here in white at the top. And so we now actually know where our SYNGAP flag is being expressed. The next question we ask is, how much SYNGAP protein is actually being made? And to measure this, we use a technique called Western blotting. So this just allows us to detect and quantify protein levels from brain tissue. So we actually collect samples from both from SRD mice treated with either a low, medium, or high dose of our gene therapy and compare them to mice that are non-injected as well. And if we just look here at the top row, we're staining for our flag signal. And just as we hoped, we see no flag signal in our untreated mice, both in wild type and our SRD mice. But once we treat with our AAV SYNGAP1 we delivered, you can see the darker band, the the darker the band becomes, indicating that more protein is being produced. And on the right, we just quantified those bands, and you can see a clear dose-dependent increase in the amount of flag expression, confirming that the higher the dose leads to higher levels of that SYNGAP protein that we're delivering. But now if we look at that second row, this is where we're staining for SYNGAP protein itself. You can see that compared to wild type mice here on the left, you can see that our SRD mice right here show a reduced SYNGAP level as expected. So when we quantify that here on the left, those are shown in these two white bars. Our non-injected SRD mice are showing about half the amount of SYNGAP protein, which aligns with what we know about this disorder. But what's really interesting is that when we deliver our SYNGAP1 gene therapy into these mice, we don't see that full dose response that we saw with the flag tag. Instead, we see that both the mid and high dose restores SYNGAP to wild type levels. So while flag expression keeps increasing, SYNGAP expression seems to level out, suggesting there may be some type of ceiling for how much SYNGAP neurons are willing to make. This is a very encouraging result because it shows us that our approach can compensate for the loss of one copy of the SYNGAP1 gene, restoring protein levels to what we see in healthy wild type mice, right? And just one more note here, on that third row, you'll notice that our flag, which is in red, and our SYNGAP, which is in green, when they overlay, it makes a yellow. So you can see that it perfectly aligns with where the SYNGAP is actually running on the gel at the expected size. So this tells us we're producing the full-length SYNGAP protein, not just any fragments of the SYNGAP gene. But altogether, these results show that our AAV SYNGAP can restore healthy protein levels in the brain, which brings us to the most critical question, right? Does our AAV SYNGAP1 rescue SRD symptoms? And that's here, I'll hand it off over to Rong to give you the good punchline. Stop
All right,
sharing,
thank you,
she'll take
Megan.
over. Yeah.
Bear with me as I make the switch. Right, there we go. Thank you, Catherine, for the kind introduction, and thank you for having me here today. So in the second half of this presentation, I'll take over and I'll focus on evaluating our treatment's therapeutic potential in the mouse model, and particularly, have my laser pointer on, particularly in the epileptic and behavioral phenotypes. Now we have this SRD mouse model, but what really matters is the patient symptom. And we want to make therapeutics that actually help the patients and resolve the symptoms. That's why with the mouse model, we will focus on the following three disease-relative phenotypes to help bridge this gap between mouse work and human disease. Now, the first measurement we will have is the EEG-EMG recordings, which we use to measure epileptic form activity in the mouse. This helps us to evaluate efficacy against epilepsy. The second assay is a behavioral assay called open field test. It is a test we use to track the mouse's locomotor activity. And this is the test we use to help evaluate efficacies for the hyperactive phenotype. The last assay is also a behavioral assay, which is called elevated zero maze test. This is the assay that we use to measure explorative and risk-taking behaviors in the mice. This helps us to model risk-taking behaviors in patients. In the next few minutes, I will go through each of these modalities in more detail. Hopefully, we'll make it more interpretable and relevant. Now, first of all, let's start with the EEG-EMG recordings, which has been my specialty for the last 10 years. So EEG stands for electroencephalogram. It measures the electrical activity of the brain. You may not feel it right now, but your brain is constantly giving out electrical signal, complicated and also related to the function. Now, to measure EEG in the mouse, we place the screw electrodes in the frontal region, specifically in the somatic sensory cortex, due to its strong association with cognitive function and the involvement potentially with sensory processing deficits in the SYNGAP1-related disorder. The other electrode is placed in the parietal region and specifically over the hippocampus. This is because of the important role of hippocampus in learning, memory, and cognitive function, and also with a lot of reported involvement in hippocampus in the related epileptic form activity. Now, these signals are fed into a little head cap in the mouse head, where we also have two EMG electrodes inserted into the mouse neck to measure muscle activity. Now, EMG stands for electromyography. If you have an electrode in your muscle and you contract it, you'll have a huge signal. With these measurements, it enables us to measure epileptic activity and brain waveforms in the mouse brain. And with help with EMG signal together with EEG, we can distinguish between sleep and wake and also quantify convulsive seizures in this animal. Now here you are looking at example traces with the EEG recording from somatosensory cortex, the parietal region, and our EMG channel. Now this one is from a wild type or normal mouse. It may just look like two lines of thick fuzzy black squares, but this is condensed EEG signal with really normal fluctuations. There is no spiking, there is no epileptic form activity. This is pretty normal with sleep and wake states. But if you look at here, this is an example from an SRD mouse. It's quite busy. There are a lot of going on, and here we will focus on two main epileptic form phenotypes. The first one is the convulsive seizures, where you can see really large amplitude synchronized firing in the brain. And this is often associated with convulsive movement as reflected with the high signal in the EMG channel. Another really important phenotype is called interactile spikes. So each one of these little blip here is actually one individual interactile spikes with really sharp waveforms. Now these only happen between seizures or when the brain is not in the convulsive state. That's why it's called interrectal spikes. Now, why do we even care about those little spikes? Because a lot of the research has proposed that interactile spikes are associated with seizure burdens, and importantly, it can disrupt learning and memory. And a lot of work have been proposing that interactile spikes can actually be an EEG biomarker for cognitive impairment in a lot of epilepsy patients. Now we have these EEG recordings we have conducted in both wild-type treated and untreated SRD mice. But what we need are numbers. We can't just show traces for any scientific findings. So what we do is we count the numbers of spikes and seizures across the full length of our recording, which is shown here as a distribution plot. So here shows over 7 days of recording, and the blue line is the number of interactile spikes in the parietal channel. So the higher the blue line, the more blue you have, the more spikes. And each red bar responds to a GTC seizure. So the more red bars you have, the more seizures the mouse is having. With this kind of distribution and quantification, the metrics we use to help us quantify the efficacy in epilepsy are the average numbers of interactile spikes and the average number of convulsive seizures. As you can see, the normal wild-type mice, no spikes. On the contrary to that, the SRD mice have over 100 spikes per hour. So that's a very robust and stable phenotypes in these mice. At the same time, for convulsive seizures, wild-type mice, no seizures, which is as expected. But the SRD mice, only a small number of them actually showed convulsive seizures during the recording, which makes it really hard to use these phenotypes as a matrix to evaluate efficacy. So as a result, our study will mainly focusing and be using the interactile spikes as a robust, consistent, and stable phenotypes for us to evaluate the efficacy in epilepsy treating. Now, the next assay, as I mentioned earlier, is the behavior assay called open field test. Basically, you put the mouse in an open box without top, but tall walls, the mouse won't get out. And once you put the mouse in, they will be starting moving around, exploring the box. And with video tracking, we can keep track of exactly where the mouse is going. We can get the distance they've traveled, how fast they've been traveling, and all kinds of parameters to help us to quantify their locomotor activity. And here, the matrix we are reporting here are the travel distance. So the wild-type mice in this hour test will run about 20,000 centimeters. On the contrary to that, the SRD mice are really hyperactive. They have almost double the travel distance. Basically, once you put them in, they will be moving constantly. Now, the next assay is called the elevated zero maze test, which we use to evaluate risk-taking behavior. This is the practice where you have a circular platform really elevated from the ground, which makes it a little bit scary. And then on the runway, you have two sections that are enclosed with really tall walls, which we call closed zones, and it is considered to be safe, sheltered, protected for the mice. They like to spend time in these enclosed zones. On the contrary to that, you have these two exposed zones, the open zones, where there's no wall. You can imagine if you're walking on that, it will be a really narrow bridge with no railing on a cliff. So it's kind of dangerous and scary for the mice, as this little buddy is kind of hesitant to work through it. Now, normally, as mentioned, the mouse, wild-type mice, they would prefer this enclosed zone compared to this exposed scary zone. As a result of that, if we quantify the time they spend between these two zones, they'll have a low ratio because they want to spend time in the closed zone, not in the open zone. But on the contrary to that, when we to look at the SRD mice, they don't really mind spending time on this scary, in the middle of the air, open zone. So as a result, you'll have a high time ratio. And this is what we used to reflect the risk-taking behavior of the mice. But in addition to just the time ratio, we also measure a behavior that's called nose poking the edge. So here is the elevated zero maze, and you can see this little fella here is actually poking his head over the edge and looking out. And with our video tracking, we're able to track the movement of their nose and quantify how many times they would just lean over and want to see out. And sometimes, rare cases, they would actually take the leap and jump out. So if we quantify the numbers of nose pokes during the whole assay, the SRD mice have much higher numbers of nose pokes compared to the wild-type mice, as another indication of their risk-taking behavior. So with all these three main assays, we will be able to evaluate our therapeutic efficacy based on these phenotypes as EEG measured interictal spikes and seizures, the hyperlocomotion phenotypes in the open field, and also this increased risk-taking behavior in the elevated zero maze. These will be our main matrix we use to evaluate the efficacy, and these are closely related to key phenotypes and symptoms in the patient population. Now, as mentioned earlier, for testing therapeutic potentials, we tried two different ages. And the first one is a neonatal injection in the mouse that's born 2 days, and we give the injections through intraventricular injection. This corresponds to the age pretty much before birth. And after the mouse reaches adulthood, around postnatal day P56, they go through all the behavior testing mentioned earlier, and then followed by surgery implantation for the electrodes, and then they went through our chronic EEG EMG recordings for a week. Now, to remind you, the neonatal treatment is where we have expression mostly in the forebrain areas, and the increase of the Syngap levels is kind of subtle, like suboptimal. Now, with this treatment, we can actually reverse the epileptic form activity. So here you're just looking at example EEG traces again, where the wild type animal, pretty normal, no spikes, just 2 lines there. Well, this SRD mice, you have the excessive interictal spiking, pretty much nonstop. And if you look at this blue shaded trace from a mice that has received our neonatal SYNGAP1 gene treatment, the number of interictal spikes are significantly reduced. Now, this is also reflected in our summary graph with the spike number and GTC numbers. So with the spike number, we are seeing a significant, although partial reduction of the interictal spikes, but unfortunately, the seizures are still observed in 3 out of the 9 recorded mice, suggesting that this treatment may not be able to prevent seizures at this age and route. And in terms of the behavior tests, I've mentioned earlier with hyperlocomotion in the open field and risk-taking in the elevated zero maze, this age and route, again, was not very effective in reducing the hyperlocomotion or the risk-taking behaviors in this mouse. Now, in contrast to that, the next, we test this treatment in juvenile mice. That's postnatal DP21 through the IV delivery, retroorbital through the eye into a systematic delivery across three different dosage from the low to the high. And again, the animals went through behavior testing around P56 in adulthood, followed by EEG surgeries and recordings. Now, remind you, this is the treatment. We were able to deliver the transgenes throughout the whole brain with really nice expression. And the middle and high doses actually was able to bring the expression level close to wild type animal in the SRD mice. Now, in this animal, we again were able to see really great and dose-dependent reduction in the key epileptic phenotypes, the interactile spikes. Here, wild types, no spikes, excessive spikes in the SRD mice. Well, here I want you to focus on this middle row with the parietal EEG, and you can see the spike numbers are gradually decreasing as we increase the dose, suggesting a really nice dose-dependent reduction of interactile spikes. Now, here you are seeing individual distribution plots. So each one of this plot responds to one mouse. Here you have 4 mouse that's not treated and diseased. And they have a lot of spikes as indicated by the blue and a lot of seizures in 2 of them as indicated by red. And then if you look at the individual plots from the low, mid, and high dose, the spike numbers are gradually and also greatly decreased, especially in the mid and high dose. You can't even see any of the blue traces really because they are so low, they're just like hovering over the axis. And non-seizure was recorded in these 4 mice that received the mid dose. So if we look at the summary plots for the interactile spikes, our mid and high dose treatment was able to near completely eliminate the interactile spikes. And then in the seizure measurement, the mid dose mice had no seizure, and then there's a low seizure recorded from the high dose. Although we are underpowered to make any statistical comparison because there was too few of them had seizure without treatment. And then in addition to just epileptic evaluation, as mentioned earlier, we can use EEG to measure brain waveforms. And this is a really important aspect of doing EEG recording. If you pass the EEG signals through something called fast Fourier transformation, which we call FFT, it can turn into a really complicated time series of EEG signal into individual spectrum components. And these individual components that we are particularly interested in are really closely linked with cognitive function. For example, the slow wave component, which is around 0.5 to 3 hertz, relates to memory consolidation, and it's pretty much the restorative power of sleep. The theta oscillation around 4 to 8 hertz reflects a key rhythm in the hippocampus. It's related to spatial and working memory and creativity. The alpha rhythm is around 8 to 12 hertz. It's an important rhythm associated with the thalamus and thalamico-cortical rhythm, and it's related to attention and consciousness. While the beta rhythm reflects sensory motor integration, cognition, concentration, and focus. While at the end, gamma rhythms, you have high gamma activity if you are processing information, if you are solving a problem, you're actively studying. So these are all key reflective properties of the brain when they are dealing with cognitive duties. Now, when we pass our EEG signal from the wild type mice, which is the light gray, and our SRD mice, which is the dark gray, through the FFT analysis, and then distinguish them into these individual components, we see different deficits across different brain waves. For example, you see increased slow wave, increased theta wave, decreased alpha, beta, and gamma waves. These are reflective properties of the dysfunction in the neuronal circuits. And in addition to that, a lot of these properties are also consistent with findings in human patients. A lot of them can be really translational EEG biomarkers in the autism and I think SYNGAP1 patient population. Now, if we look at our rescued mice, so after the treatment, in a dose-dependent way, we are seeing restoration of the slow wave, the theta wave, the alpha, beta, and gamma wave, pretty much across the spectrum. We are seeing restoration of network oscillations. But besides just network oscillations, this is also indicative that we may be able to restore the state-dependent brain functions, which are closely associated with these frequency bands. Now we are seeing great epileptic and neural oscillation rescues. Let's get to the behavior tests. So if you remember the open field test, which tracks the locomotion, and the untreated SRD mice, they have hyperactivity phenotypes, so they just travel a lot. And here, if you see the injected SRD mice, we're seeing a really nice dose-dependent decrease of their travel distance in the open field test, suggesting that we are able to reverse their hyperlocomotor phenotypes in a dose-dependent way. Next, in the elevated zero maze test, where you have increased the ratio of staying in the open zone and also increased nose poke numbers. And you can see we are also getting a nicely dose-dependent reduction of these behaviors and these measurements, suggesting that in the true mice, we're able to reduce the elevated risk-taking behavior after the AAV gene treatment. So to wrap up a little bit and to make a good summary of what have been found so far in terms of the in vivo efficacy testing. Now, for our neonatal ICV treatment with kind of forebrain biased expression and suboptimal expression of the gene, we have partial reduction of the interactile spikes, but the seizures and any other behavior phenotypes we have are not really rescued. On the contrary to that, with our TrueNL IV injections through the opto route, we are seeing really nice brain-wide expression, dose-dependent restoration of the Syngap protein. In these mice, we see dose-dependent reduction of the interactile spikes, particularly the mid and high dose was able to completely eliminate the spikes. The seizures were underpowered to make a conclusion, but we are seeing really nice dose-dependent rescue for the hyperactivity and risk-taking behavior. So at this point, I'll hand it over to Megan and then help us to conclude and project to the future directions.
Yeah, well, thank you so much for that great talk, Rong, on getting us through all the behavior. But today I hope that all of this taken together, this work represents the first preclinical validation of an AAV-based gene therapy for SYNGAP1-related disorders, right? Today we showed that we successfully were able to develop a functional AAV SYNGAP1 construct. We demonstrated that it's specifically through the intravenous delivery, we were able to get broad brain-wide expression and elevate total Syngap protein levels to wild type. And most importantly, what Rong just showed you, that this therapy can rescue epileptic and activity and improve behavioral outcomes in a dose-dependent manner. So that's pretty important. So this provides us with a strong proof of concept. But of course, before turning this into a therapy for patients, this requires the next phase of work, right? Bridging from our animal model to potentially going towards a clinical translational work. But I think I wanted to be, it's important to be clear that what we're showing to you today is just a proof of concept in mice. It's not yet a therapy for patients. There's still a lot of work that needs to be done before we could consider moving this into human studies. Specifically, we would need to first optimize and define the final clinical candidate vector that we would use, right? The version of the AAV Syngap we used in the studies is just for preclinical testing. We would need to refine the vector and the capsid that we would use to ensure it's as safe and effective as possible. We would also need to determine the therapeutic window, both age and dose. So we need to understand the optimal age. We just tried 2 ages. And we would also want to more finely tune the precise dose required to achieve the therapeutic benefit window. And then finally, but most importantly, we need to thoroughly assess the safety profile because safety is paramount before you give it to humans. So we need to make sure that we're delivering a therapy that doesn't cause any unintended effects of the brain or other organs. So while this work represents a major first step, it's still very much in the preclinical stage, and additional studies would be required before any human trials can begin. And with that, I would like to take a moment just to acknowledge that none of this would have been possible without a huge team effort. That's what the Allen Institute is known for, is big team science effort. So this work represents a contribution from many people, and I'm grateful to work with each one of them. Just a few special shout outs. I have Boaz Levy and Ed Lean, so they led and guided the overall direction of our research. Andrew Clark, he handled all the mouse tissue processing and was critical part of the work that allowed us to see what was happening in the brain tissue that I showed earlier. Em and Robert, they performed the EEG and behavioral analysis, helping us understand how the therapy affects the brain function and behavior, which is so important. Ref, Aaron, and August, they helped make all the AAV constructs, and they tested their function in cells, ensuring that the therapy was working as we expected. And of course, Catherine, our project manager, who coordinated all of the moving parts in our meetings and everything. And of course, there are many other team members whose efforts were essential to this work. So I hope this project truly demonstrates that we're a dedicated team, that we were able to accomplish this common goal of developing a potential therapy for SRD. With that, we'll take any questions.
Wow, thank you so much. While you were talking about all the people that contributed, could you also give us a sense as to how long you were working on the project?
Yeah, that's a great question. I think it's about 3 years. So when I met 3 or 4 years, when I met you, I literally had just joined the Allen Institute like 2 months before. I think I joined in September of 2022.
Yeah.
So
Yeah. So I just wanted to point out how many person years that was of work. It's a lot.
yeah, a lot of work. It was worked a little on before I got there too, but it's probably about 4 years worth of work that we were able to do for this project.
Yeah, wow. Thank you so much. I am going to go to the Q&A, but first I wanted to ask a couple of kind of more technical questions, if that's okay.
Of course.
So for wrong, I was wondering about the time of day that you did the behavior testing. So the time of day for the mice, I guess they're nocturnals, but I don't know which, if you switch them so that you can do your, if their night is your day, or if you, do you see what I mean? So were they, what was it, day or night for them? Was it when they're active? I guess day and night's the wrong word. Is it when they're usually active, or was it when they're usually inactive?
So for this assays we have, I think it's during their inactive phase. So they would be in their light phase.
In their light phase. Okay. And so and so the wild type mice do some amount of activity then, even though they're usually asleep, even though that's usually their inactive phase?
So even though it's called active, inactive, mice are polyphasic sleepers. They don't sleep or like solid period of time.
Oh, I see. Okay.
Yeah. So even though when they're totally like should be sleeping in the inactive phase, they still wake up and move around a lot. There is no like continued sleeping, which is one of the challenges for people to do sleep study, which I've been doing for the past like 7 years before I joined Allen. Because they have really fragmented sleep. Their episodes of sleep is like 20 minutes or so compared to our 8 hours. So even though during their inactive phase, in the light phase, you can still do behavior testing. If you're looking for something that's like really attention seeking or something, I would recommend to reverse it so that they are actually in their active phase, in the dark phase. Yeah.
Okay, great. Thank you. And then for Megan, with the expression in the mid and high dose, so this would just be in the juveniles, you showed us pictures. Can you tell us about, you showed us pictures and showed us how the protein expression kind of leveled off, right, between mid and high dose. Can you tell us anything about the spatial range of expression between mid and high? So did you see dots? Like, I don't think you showed us the sagittal section of the dots, right?
Oh, of the mid, yeah. So you can kind of by what you were seeing when you're looking at the brain section, that's immunohistochemistry. And while you can measure like amount of protein that's actually getting expressed in that, it's a little bit harder to do, and why I prefer doing the Western blot, where you're just looking at the bands to see if you're getting, like, it makes it much easier to quantify what levels you're actually getting. But by immunohistochemistry, we can also see that there's a difference between the doses as well, but we don't see any difference in terms of like how widespread it is. But it's definitely harder at the low dose to see the expression by in that brain section to see any expression. You have to really turn up the
the signal, yeah.
the signal in order to see it.
Yeah, yeah.
So it's a large protein, so it's hard to express sometimes. So I think we were
Right.
pretty
So
excited.
yeah, so the thing I was thinking about was with the early dose, you can only look at low because they're so small, and so there wasn't good rescue of things then. But so I was trying to understand if it had to do with the total dose or just not getting to the right place, or I guess we just probably won't know that. But
yeah, we don't know that, and it is an important point, right? We did only pick one dose for the P2 ICV, but in theory, in a perfect world, we would have done a larger dose range study, and we can go a little bit higher, but when we chose that dose, the 1E11, that's a very typical dose to give at that age, and that's where we stopped. But there are ways you could give more. You could go a little bit higher as well to see if that's it, or is it the difference in how that route of administration gets a little bit different in terms of like where it's actually getting expressed, right? We didn't do the IV delivery in the P2 mice. We did the intracerebral ventricular. So it gives a different pattern. Or is it the age? So they're not quite directly comparable, but I think
that makes sense.
We were pretty excited to see that even with that like subpar expression of SYNGAP protein, we were able to see a partial reduction in the intracranial spikes, and why we wanted to share that, right? That was pretty exciting to show that, but we don't know why we see the differences. There are a number of reasons and definitely of interest.
Yeah. Thank you so much. And then I'm going to ask you one more question that kind of gets into something that Suzanne Jones asked. She's asked the second open question in the Q&A. So when you showed us the Western that had both the pan Syngap antibody, is that what you called it? Pan Syngap? Anyway, it was both types of Syngap, right? It's both the endogenous and the added, the AAV delivered Syngap, right? It's a combo. It shows them both. And then the flag tag. So then you can see that overlaps. The flag only overlaps the combo. So what I'm wondering about is if the flag tag can you tell if there is more of the flag tag in the high dose than the mid dose? And can you tell what is being depressed? Is it the endogenous being depressed, or is it both, or is it, I guess that's my question. Can you kind of tell what's happening there?
Yeah, that's a great question and something we're definitely interested in. I think ways that we could probably look at that. Thank you for bringing it up. And I was actually going to mention, if you went to at the end of the slide deck, I actually have like the full Western blot, so you could see what it looks like, right? Because we're blotting these at the same time. But I can't tell here. You can see though that the Syngap does look a little bit different in the wild type non-injected, right? You're getting different bands, and by pan Syngap, it's just an antibody that actually detects all the different isoforms of Syngap. So there are specific Syngap antibodies that will recognize specific isoforms, but we're just using the one that recognizes all of them. But what I think you can see here and hopefully appreciate is that our Syngap is really changing how it looks, right? This one has like a darker band down here and maybe a lighter band up here. And then here, it's kind of all from this Syngap that's getting expressed most likely from our flag, but we don't know that for sure.
I think since Wrong is sharing, we're not seeing your.
You can't.
You want to share? Like, you can pull it out.
I forgot.
We'd have to have Wrong doing the cursor.
Yeah.
But I think I know what you mean. Like it's going to be hard to pull all that information out of that Western. But that's an interesting question. And can I just read what Suzanne's question was then?
Of course, yeah.
Okay, so her question was, it would be interesting to learn if Syngap protein is stopped by the body from being made in excess, as you just mentioned, with missense mice. Is that something you can study? So I was wondering if with the AAV, if that would maybe push down the endogenous with the and have more of the AAV based one.
Yeah.
But because we're not going to be sure. I mean, maybe it would differentiate between wild type in the sense, but I'm not sure why it would. It seems more like it would maybe depress both of them, or not.
Yeah, if I were to guess, you're just depressing the endogenous, but we don't know that for sure, right? We would need to test that empirically. But those are definitely interesting questions. I don't think it's what we expected, was to see this ceiling effect. But I just don't think we expected to see that.
Okay, so I'm gonna skip Beata's immunity one, because I think this is more of a, unless you guys want to, I mean, have you guys read it? It's the first question. If you guys have a good answer for this, that's fine, but I wouldn't expect that from you necessarily, since this is preclinical work. So do you feel like addressing that, or should I give
say something? I can say something briefly. I think it is definitely a point that we consider in the AAV gene therapy space, right? You would get tested to see if you have been previously exposed to antibody or
okay. I'm not sure everyone can see it, so let me just say that Beata's asking, can you please explain how immunity to this vector, which is the AAV, works? And then do you check antibodies for the virus in the patient before the therapy? Do they go away after a period of time? Are there patients who would be disqualified for a viral vector in general? Is this kind of therapy given once in a lifetime, or possibly be repeated? If it's given and doesn't work, can the patient get this kind of vector therapy again, or is it one? So anyway, those are the kinds of questions.
Yeah, and this is all heavily being studied in the AAV space. And I know there's actually a recent work, so it's not in human yet, that they're trying to say if you've been previously have antibodies against AAVs, they can try to reverse that, but that still needs to be studied way more before it gets to patients. And the idea is that ideally this could be given one time, but we don't know that for sure. There could be a case where you might need to redose later too. So that we want to be careful when we're talking about AAVs, right? It could be dependent on what you're given. So that would be way past where we're at right now. But for the mice, we gave it one time. And then we saw effects in adults, right? We gave them early and then measured them when they were adults, so much later in life.
Right. So I want to point out that this is a SYNGAP specific talk, and that those kinds of questions would be answered in a big AAV talk. So that's just for other people to hear that it's not really in the scope right now, but thank you for giving us that information. Okay. So another question from her is, do you see the need for age cutoff for this therapy? I think that's because you did too, but you already kind of addressed that in your.
Yeah, we would like to try later ages for sure to see how far can you push it, because we already do know that you can, like, seizures are definitely rescued later in life through other genetic mouse models. So we would want to see can we give it later, and does it still rescue?
Right. And so I'll just skip down to anonymous attendee, who also asked a little after 5, how long before we can test timing of the dosing at P70 adulthood?
Yeah, you could do that as soon as you would want. Yeah, as soon as we can inject the mice and get them at P70. Yeah, that's not in mice, that's not hard to do.
Right. I think one thing that we haven't talked about too much, though, is that this project is now complete for right now.
Correct.
So do you or Rong want to talk about that? Like, is there anything, Rong, are you still doing EEG data analysis on it, or is that wrapped up?
For Syngap, we still have a lot of data, but I'm not really sure like at which timeline or what part of this, like the additional data we should like do with it. And in terms of the future experiments, there's a lot of uncertainty. We're certainly looking for ways for us to continue. There are a lot of interesting questions we really want to answer, particularly the adult rescue, because not for this disease, for some other ones I'm working on, we are seeing great rescuing adult mice, adult treatment. So that will be really exciting if we can do the same thing. But again, we are looking for resources and try to continue the great work.
And when you say other disorders, I assume you're saying other monogenic neurodevelopmental disorders. Yeah. So something along the same lines. Yeah. Okay, and Kathleen Malloy also asked what time frame is feasible for this therapy to finish animal testing and move to human clinical trials. I think this project getting picked up by getting picked up or funded by someone is really the important bit.
Correct. Yeah.
Yeah. Okay. An anonymous attendee, could this provide rescue for a pathogenic variant in the first 4 exons?
Yeah, it's a good question. We would definitely want to test this now, not just in a specific loss of function model, but if you could see if this would also work for, say, specific point mutations, or would it work in other animal models as well?
Yeah.
Right now we don't know.
But you also use the entire gene, so this theoretically this would work. I mean, yes, you're right, you would want to test it, but the theory is that yes, it would work. Is would be my answer. Is that
correct? And yeah, especially if it leads to, our idea is that if it leads to a loss of the SYNGAP function or protein, and in theory this should work. We're just not sure if you have a different mutation that doesn't lead to a loss of the protein itself, would this work? But yeah, we would want to test that for sure. There are other mouse models that I know people have made, and they look great, so we would love to test those.
Yeah. So I have another question here from Olga. Is there a way to measure aggression in mice? It's a big component in SYNGAP1.
I wouldn't call the hyperactivity as like aggression, but there are specific behavior assays in the mice we can do for aggression testing.
So the answer is and then she says, sorry, let me so that one's done, or is hyperactive. Yeah, so it's not as close as you can get. There are other things. And then I don't know if Boaz had any thoughts or questions he wanted to say anything to wrap up. Boaz is someone I got to meet in person also. He also showed me around at Allen Institute, and he's the leader, he's the boss, he's the director. I don't know. I'm not sure which words I should use, but it's the guy in charge. Okay, so he says no comments. Perfect. No comments. That's right. So I just want to finish with saying thank you. And I'm not, and Suzanne, I'm going to read your thing. That's part of my finish, so don't worry. But I want to finish by saying thank you so much to both of you. I've been so impressed with both of you and all the hard work you've done. And when we've talked, at length privately, it's just amazing the amount of effort and thoughtfulness and really just sort of scientific resilience you guys have put into this project. One thing that we used to hear a lot around SYNGAP therapy, when SYNGAP therapies were in development, was it's really complex. And you guys didn't really come in saying that. You said, yeah, it's complex, but we figured it out. And I asked you, how did you do it? And you're like, well, we just kept going, and I was like, why did you keep going? And you're like, well, we thought we could, and so it really shows how this big team of people really working hard together over many years, like how that actually moved the needle forward on this. I want to make sure people understand what a big deal it is to see these comprehensive changes in expression, in EEGs, and in behaviors. That's 3 really separate things that they're looking at, and it's all really interesting. So with that, I'm going to read what Suzanne says. This is such exciting work. Thank you. And what a fantastic presentation and slides. Agreed. We greatly appreciate you taking the time to document and explain your work to non-scientific people and parents in a way that is understandable. And I agree, you guys do such a great job of having the detail and the words, but also, for people who really want to know what's in there scientifically, but also saying it in a way that's accessible. So if you want to skip over some of the words that are a little more strange, we still understand what's happening. So thank you so much. I'm really, really impressed with you all. Oops, I'm gonna type, I want to answer live and done. And I feel like there's still, it was just that one. Okay, we did it. I think I did it as host, which I'm not usually host. So
did a great job.
Yeah, and really thank you for having us here. I think it's a great opportunity for us to show our work, and we are really excited like to talk to parents and be able to make our results interpretable in a way like to a broader audience. That's really exciting.
Yeah. Yeah. Thank you so much. Okay. Well, take care, everyone. Bye.
Thank you. Bye.